Thank you. I'm very excited and grateful to be here. Like they said, I will talk about modeling theta phase-dependent sequence memory with recurrent neural networks. My name is Matthijs, and I'm presenting work conducted jointly with Stephanie Lobe in collaboration with Johannes Niedek, Florian Moorman, and supervised by Jakob Macke. I need to get my slide. So if you think about one of your favorite movies, you can probably recall several scenes, and not only the contents, also the sequence in which these scenes unrolled. How does the brain represent the sequence of information? We might want to look to the medial temporal lobe, and that's because of two findings. First of all, we can find cells like these. The cell pictured here seems to fire strongly when presented with the notion of Luke Skywalker. These kind of cells, termed concept cells, have previously been hypothesized to form the building blocks of episodic memory. At the same time, in these regions, we can find that theta oscillations tend to synchronize during memory tasks. Think about encoding or retrieving information. A way we could hypothesize these two findings to come together in a form of memory of sequences has been proposed already in 1959 by Lisman and Idiart. The idea here is that if we are attending a sequence of, for instance, 3 letters, as the person in this picture, then we can find single neurons coding for each of those letters, and the neurons coding for these will fire one after the other every cycle of the theta oscillation. The order of the letters in the sequence is then maintained by the order in which the cells coding for them fire within one cycle of the theta oscillation. So do we find this kind of phase of firing coding in human single unit recordings? The short answer seems to be yes, but the long answer seems to be not entirely as we expected, but in a way we can capture with recurrent neural networks. And then we can use these models to further investigate what's going on. I will first talk about the experimental approach and then talk about the recurrent neural networks. Single unit data was recorded while human participants or human patients performed the following task. They were shown a sequence of 4 stimuli, one after the other, which was followed by a delay period, after which a stimulus panel comes up. In this stimulus panel, we can again, in every row, see the same 4 stimuli, but possibly in a different order, possibly in the same order. The task for the human participants is then to pick the row in which the order matches the initial order. So the right answer for this trial, it will be the top row. In total, we had access to data from 13 patients, containing in total over 1,000 single unit recordings, from which we picked 217 stimulus selective units. So how does these stimulus selective units look like? Here we can see example spiking behavior of a cell at which a particular stimulus, in this case the Queen of England, was presented at a time corresponding to this vertical black bar. We can see that this neuron or unit clearly increases its spiking rate right after the stimulus is presented. When we look at the neuron's firing rate after any of the other stimuli during the trial are presented, we can see that its spiking behavior remains near baseline, or close to baseline, indicating that this cell is strongly selective for the stimulus Queen of England. Now remember that within one trial, a sequence of 4 stimuli is shown. So a neuron's preferred stimulus, for instance the Queen of England, can be shown at sequence position 1, 2, 3, or 4. So we can group trials by the position a neuron's preferred stimulus was shown at. So here we have trials on which the Queen of England is shown at the start of the sequence, and we can see a clear increase in firing rate again after stimulus presentation. When the stimulus is shown at position 2, again a clear increase, 3 again, 4 again. So independent of the position a neuron's preferred stimulus is shown at, there's an increase in spiking behavior. We can get these continuous traces by simply convolving the spikes, and we can then sum up over all stimulus selective neurons on trials in which their preferred stimulus was shown. And we can again see that there's a clear increase in firing rate right after stimulus presentation. We can also see a clear increase in theta power, around 3 to 4 hertz, during the delay period of this task. Now we can look at how stimulus selective neurons fire with respect to the ongoing theta oscillation during the delay period. let us briefly remind ourselves of what we would expect based on Lisman and Idiot's model. If we have a trial on which a neuron's preferred stimulus, for instance, Queen of England, is shown at sequence position 1, and then followed by 3 arbitrary stimulus, we would expect a neuron coding for this stimulus to fire early on every cycle of the theta oscillation, as it's pictured here. If the stimulus is shown at position 2, we would expect it to fire a little later, and so on. Now let's look at actually how our neuron's firing rate looks like with respect to the phase of the theta oscillation. Here we see the proportion of spikes as recording from a neuron as a function of the phase of the theta oscillation. We can see first of all that the neuron phase locks to the theta oscillation as it fires mostly at a particular phase, as we have a non-flat distribution. We can also look at actually the phase it looks at, which is around 0, and the mean we can see with this dot here. Now let's look at the same plot, but now the neuron's preferred stimulus is shown at position 2 to 4 in the sequence. We can see that the phase at which the neuron likes to fire is different based on the position its preferred stimulus was shown at in the sequence. However, what is unexpected is that it does not seem to be the case that the neuron fires early on in the theta oscillation if the preferred stimulus was shown at position 1, then a little later if it was shown at position 2. Instead, we can see that this neuron fires maybe early on in the theta oscillation when its preferred stimulus is shown at position 1, or 2, then 4, then 3, then 1. So instead, we have something like this. In fact, we have many such units in our recordings whose phase order does not directly map onto the stimulus order. We can count for how many neurons the phase order does not follow the stimulus order, and we can see that this is the case for most neurons we recorded. So a quick recap of our experimental results. We can find these single cells preferentially firing for a certain stimulus, and these neurons phase lock to ongoing theta oscillation. However, the phase at which they lock is not entirely as would be predicted by Lisman and Idiot's model. So what about recurrent neural networks? When we train them on this task, will we retrieve the same behavior? And do we then have a model that in the future we can use to better understand what is going on? We trained RNNs to perform a multi-item short-term memory task, mimicking the original task performed by the human participants. The RNN, the recurrent neural network, was given 8 input units, each representing one of the stimuli in the original experiment. 4 out of those were activated during a particular trial during the stimulus period. Then, after a delay period in which these and their order had to be maintained by the network, again, 4 stimuli are activated, but possibly in the same order, possibly in a different order, which the neural network had to answer. It has asked whether this order matches this order. We imposed biophysical constraints, such as Dale's law, and we optimized the weights and time constants using backpropagation through time until it could successfully perform this task. Again, we find stimulus-selective neurons, also in our recurrent neural network. Here we can see example mean firing traces on trials on which a neuron's preferred stimulus was shown at position 1, 2, 3, or 4, and we can again see a clear increase in firing rate on stimulus presentation. The spikes here are simply added for visualization and drawn with a probability proportional to the continuous firing rate. We can again sum up the traces of all stimulus-selective neurons on trials in which their preferred stimulus was shown, and normalize this, giving us again a clear increase in firing rate and the stimulus presentation at time corresponding to these dashed lines. We can also see clear oscillatory behavior during the delay period of this task, even on average. Where we calculate the model's local field potential by taking the absolute synaptic input to neurons and then taking a power spectrogram, we can see a clear peak at a low frequency. We can tune the oscillation frequency used by the model by a newly developed regularization term that we can add to the loss, which promotes power at a user-specified frequency. We can again look at which phase a neuron likes to fire during the delay, or in this case, at which phase neuron has the highest continuous firing rate during the delay period, as a function of the position its preferred stimulus was shown at. We can again see that neurons tend to have the highest firing rate at a particular phase during the delay period, as again, our distributions are non-flat. We can see that the phase at which a neuron likes to fire is different depending on the position its preferred stimulus was shown at, as the location of the peaks is different. And again, the phase order, which in this case might be something like 3, 1, 4, 2, is not the order we expect, because we would expect it to be 1, 2, 3, 4, like the stimulus order. And just to say, we count circular shifts of the same order to be the same. Again, we can count how often the stimulus order does not map on the phase order, and this is the case for most neurons, also in the RNN. So as a quick recap, we find stimulus-selective neurons whose phase of firing depends on stimulus position, but it doesn't follow, the phase order does not map entirely on the stimulus order as we would expect based on Lisman and Idiart's model. And we found this both in biological units in the human medial temporal lobe, as well as in our artificial recurrent neural network. And here, I want to emphasize that this is really an emergent property in our network. The oscillations were not put in, but just simply arise and use this phase coding mechanism after training. So as an outlook, we are currently investigating some model parameters, seeing if the phase ordering we find during one cycle in the delay period depends simply on stimulus offset, oscillation frequency, and also whether having an input-driven oscillatory input also leads to similar or different behavior, as right now the oscillations emerge autonomously in our network. We are also looking more into general role of limit cycles in memory processes in RNNs, and have, for instance, been able to show that a continuous valued stimulus amplitude can be maintained in the phase of a limit cycle that we can find in simple RNNs. So finally, I want to thank Stephanie Lobe, who conducted the first half of this analysis, Jacob Makker for supervision, Johannes Niedek, Florian Morman for data collection, Omri Barak for further suggestions on the dynamical systems point of view, and Richard Gao for further useful suggestions. Thank you all for listening.